So first of all, I would like to thank organizers for giving me the opportunity to present our research and our lab. And actually, we are CZ-OPENSCREEN, National Infrastructure for Chemical Biology. We develop and we look for small molecules as a tool for manipulating different biological processes. And these activities involve high-throughput screenings of small molecules and chemical libraries. So we are academic lab, so we are based in academia. Here you can see the small team of people, although this is not really up-to-date photo. The main people of this lab are there, and I would like to stress mainly these two guys, like Ctibor Škuta and Tomáš Miller. These are our software developers, and those guys are responsible for the development of software I will be talking about later. So we are based in the south of Prague in the Life Science Institution campus. This is our mother institution, Institute of Molecular Biology, Genetics, sorry. And this is our lab. We are at the first floor of this wooden building. So the mission of CZ-OPENSCREEN is the identification and development of new molecular probes for research and new potential therapeutics. We've got quite good funding now from Czech government, which allows us to do our own research. But in the same time, we are also responsible for providing certain services to scientific community. And there are actually three areas for our services. There are three types of users. So the first type of user are biologists. So with biologists, they can come to us and they come to us with their own biological problems and their own research. And often they want us to develop a selective probe for manipulating and studying their biological problems, research. And we talk to each other, and we think that the project is sound science, if it is really interesting and high quality, then we start working together on the development of the project. And eventually we submit it to our open access committee that evaluates the project. If there are some issues, it comes back to us, then we rework it, resubmit again. And when this project is mature, then we start working on it, and we start developing assays for the primary screen, assays for the confirmatory and validation phases, and also all plans for medicinal chemistry, hit-to-lead optimization, and so on. Then there is a second area of our users. Those are chemists. And actually, what we do for chemists is that they can bring their compounds and put it on our deck for high-throughput screenings in really interesting biological projects. And on top of it, we also provide basic characterization and profiling of these compounds using, for example, we can measure cytotoxic profiles on a panel of cancer cell lines. We can check for the interaction with microskeleton antitubulin compounds. We can identify compounds which somehow interact with cell cycle and so on. Chemists then take this information, they can find some inspiration in that, or they can publish it, they can do whatever they want to do. The good thing is that if they really put their compounds on our deck, then if these compounds or some of the compounds is found active in the screen, it might be really a nice start of collaboration, and it can be motivation for chemists to do so. Finally, the last area are data users. If you do a project in open access, mode, then we are supposed to open the data to the public, to everybody after 3 years. Unfortunately, this is still not running totally due to some technical problems, but we plan to open and release first data this year already. And also what I wanted to say is that there are two modes how we deal with our users. So either the user can come and we can do our work in collaboration. So in that case, we actually cover the most of the costs of the project, and then we share authorships and IPs, or we provide these services on the basis of fee for service. Then we don't ask much and simply deliver measured data. So this is a basic overview of our technical equipment. So we've got two automated, integrated robotic stations with robotic arm inside and different instruments around. We've got plate readers, we've got automated microscope there. We are basically able to measure every possible readout, like fluorescence, polarized fluorescence, time-resolved fluorescence, alpha screen luminescence, impedance. We have experience with most of the assays around. So we do protein binding assays, enzymatic activity assays, cell-based functional assays, also cell-based phenotypic assays and label-free assays. And what is important to say is that the compound transfer is done using the technology from lab side. So we use acoustic compound transfer, which is a really great thing because it gives you really the flexibility of creating really complicated assay layouts, and there is no risk of contaminating your compound library during the screening. So this is a great advantage. And also, finally, the most of the assays we run in the lab are run in the 1536 format, which shows also the our experience that we can handle such data, which are prone to many errors and artifacts, but most of the assays are done like that. Well, our current compound library consists of, or includes 85,000 compounds currently. The most of the library is formed by so-called diverse set, the diversity set, containing 70,000 compounds. They are drug-like molecules from different commercial sources. Then we've got also a collection of bioactive compounds, including well-defined and selective probes, and also some drugs. There are natural products, and then we've got unique compounds from not only Czech chemists, but mainly from academia, from our country and surrounding countries. And finally, there is also a really nice collection of steroid compounds, which includes about 4,000 different steroid molecules. It's really unique. It's from IOCB from Prague. Well, and this is a small overview of projects that we have done recently in the lab. So we've got a couple of projects in the area of DNA damage repair. We had a really interesting and unique project, long-lasting also, for identification and development of modulators of microRNA pathway, and also a key component of this pathway, which is a dicer molecule. Then we do a lot of pathway profiling, like Wnt, sonic hedgehog, hypoxia-inducible factor. Really interesting project that we started recently is this de novo purine synthesis pathway, which was started. The main motivation for this project was to develop compounds that would help patients with rare hereditary diseases. And if we are successful, these compounds can also be used in the treatment of some oncological diseases. Also, I would like to mention something about our own research. So this is the area where we are quite unique because we have developed a large platform for profiling of nuclear receptors, and anybody who is interested in this area and some diseases related to this area can come to us and can use our dozens of tools for these receptors. Okay, so when we started our lab and first high-throughput screenings, it was 10 years ago. We started really from scratch, and we had really very limited experience with high-throughput screening. So we started with just pipetting robots, and there was no integration at the beginning. We learned step by step everything about this business. And also, we didn't have any software to analyze our data. So we were looking for some options, and we were quite disappointed because all the solutions we saw at the time were quite expensive. And sometimes, even at the time, they looked already outdated, and we didn't really find the motivation to go either way and buy some license. So we simply decided that we would develop our own LIMS system and all tools necessary to analyze, to run our projects and also to analyze our data. And this is where we are now. So these are screenshots from the ScreenX, our LIMS system, which really supports our research in the lab now. And it's a web-based solution, so it runs on a remote server, and everybody in the lab can connect to it using browser. From the technical, from the background, I can only tell it runs on PostgreSQL. There's a RDKit behind it, and last year we purchased license for Marvin.js, which is slowly implemented in the LIMS too. This is all I can say because I'm a biologist, and this is what I understand. And currently, the system allows us to really take care of whole compound management, sample management, plate management. We also design our experiments inside these LIMS. We store data, we analyze data, we report, and there's also some basic cheminformatics intelligence embedded inside. So this is the view of the library. Here you can see some of the compounds in the library. You can see the structure. Then there are some links to public databases like PubChem, Campbell, Zinc, if they exist. ChemSpider is there. There are also links to all samples because the compound is virtual, and only samples are really things that exist somewhere in freezers and which can be screened later. We also generate some or calculate some properties of these compounds. Of course, you've got the library to be screened in an assay. So this is already, you take the library, you run the screen, you collect data, and you store the data in the database. And we developed tools to analyze the data later on. And this is the view of the results from the primary HTS. This is small HTS, including something like 14,000 compounds. You can see the activities here. And this is actually the moment where you can also somehow clean the data, normalize data. So we can do the normalization inside the LIMS. Of course, you can run a normalization like fold, activation, percent activity, and so on. But on the primary HTS data, we run a complicated iterative algorithm, and we calculate Z-score or B-score of every measured sample. And this is something you cannot do in Excel, so this is done internally. And this normalization removes all possible artifacts like interplate, intraplate variability, edge effects, row, column effects, and some problematic areas. You have also some basic statistics of the primary screen and some tools how to identify your hits. This is alternative view. Here you can see the activities of samples on plates as they were really measured. What is important to say is that this is interactive, so you can actually go from this graph, you can click on the hit and you see its position on the plate, and you can decide whether this is a specific activity or is it in some area where there are many problems. You can exclude it later, and so on. Well, once you select your hits, you take your hits and confirm the activity either in the single dose or in dose response. For dose response experiments, we have developed quite a handy tool application that really allows you to create very complicated dose response experiment layouts. It allows you even to make combinations of compounds in dose response, one on top of each other. And I think this is really useful, although this was designed mainly for echo device. We keep in mind that there is acoustic transfer behind it, which can pipette everything, actually virtually everything. But it can be adapted to some other dispensing solutions too. Well, you start with selecting your samples on a compound plate. You can have many plates with thousands of samples. You select them either on the plate directly or you enter them as a list of samples. And then you start designing your dose response experiment. So the starting concentration, what is your step of for the serial dilution can be any number. The final, the lowest concentration is then calculated. You also you can enter dose response point count, so how many doses you are going to measure, how many multiplicates, and many other things that affect finally the pipetting. You've got also the option to design the experiment on 96, 384, 1536 well format. And once you are done, you simply fill the plate or plates with your samples. It can look like that. There is a heat map, so lower concentrations are brighter, darker are more concentrated samples. And you see that you don't really use up efficiently the whole space in the plate, so you can make some adjustment, and the experiment can be performed in a better way like that. There is another option, finally, that you can scramble all your samples just to avoid, some systematic artifacts that affect one specific sample. So the experiment can be then performed like that and restored after the measurement of your data again in the limbs. So actually, this is our application for dose response experiments. I think it's can be inspiring for other people who do this kind of experiments too. Okay, then you measure your experiment, you get the data from those response experiments, and again, we've got the application inside the limbs that allows you to analyze the data. Here I would I would like to stress that this is again a web-based application, and although we analyze hundreds to thousands of samples and those responses, this is still pretty fast, and it works really very nice. So in this analysis, we normally first filter out samples which were not active. So we have a cascade of parameters that helps us to identify inactive samples, and those samples which were active, they are then fitted with a dose response curve, and obviously all samples are first fit using this algorithm, and then you can work on individual sample even on this web page, which I think is a really marvelous thing and feature of this application. So then you can tweak every specific curve that is not fit correctly. Then, if you calculate your IC50, you forget about doses, about these curves, and you calculate your IC50s and EC50s, and we also are quite busy on developing tools for the analysis of multiple assays, because in this process you need to actually collect data from really very different experiments. Some of them are counter screens, some of them are screens for the mechanism of action of these compounds. So you need to collect a lot of related information about every sample, and you need to analyze them. And especially if you have really a lot of compounds, that it can be really challenging task. So in this case, this is our solution. Every sample that was analyzed in 5 different experiments, so these are IDs of the experiments, are listed here. And for every sample, the activity data are listed on the row. So the first view of your data is on this heat map. Sometimes it's better to look at the heat map than on the curve, but you've got both options. And again, this is a dose response, so you have lower concentrations here and high concentrations here, and you can easily see that, for example, this compound has really clear activity in these 2 experiments, probably this one, while it's weaker in these 2 experiments. Then you can also check curves and calculated IC50s. If you are done with this analysis, then you can cluster your samples according to these IC50 values, or according to the structure, or you can do both. So you've got the graph with clusters of structures and activities in the same time. And again, this graph is interactive, so you still work on activity data, and you can zoom in, you can zoom out, you can really investigate in a detail some specific cluster of compounds from the point of view of the activity or from the point of view of the structure. If there is some common scaffold, you can see if this is also reflected totally in the activity data. To be honest, this part of the LIMS is still under development, but we use it quite successfully in our workflow now. Okay, so until now I spoke mainly about our own software, but since the very beginning we have been using tools from ChemAxon. We never tried to integrate them just because we wanted to keep some freedom and rely either on open source software or our own solution, but ChemAxon always made a great software that helped us and is helping us every day in the screening process. So at the beginning we are now implementing Marvin.js in the beginning of the screening. So in the compound management you can use Marvin.js for entering the structures and for dealing with structures. Then probably in the later stages of HTS, so once we have selected hits, I really like to use instant JChem. At the beginning, I tried to use instant JChem to do structural searches and combine them with activity data, but this was never so rewarding, and finally I gave up, and I found that instant JChem could be really very efficient in structural searches, while then I can export the results of my searches in separate files and then analyze them in other software for the activity. But what I do very often is that after the primary screen, we identify hits. We then search with these hits again back in the database, and we identify similar compounds there which were not active. Then we include them back in the validating experiments and build a kind of SAR already. So this is great. Instant JChem works well there. We also use JCluster. My feeling is that this tool was a bit forgotten by developers, and although this is a really useful thing, sometimes it crashes in my hand. But I really like to use it anyway. I think it would be quite interesting if somebody tries to implement it into instant JChem. And finally, in the later stages of HTS, we use JChem for Excel a lot. This is so flexible tool for so many tasks like building of SAR models, R decomposition. What is great is the functions, cheminformatic calculations available through functions, naming, conversion of names to structures and back is really great. So these are tools that we use side by side together with ScreenX LAMS. So yeah, that's all from me, and I would like to thank you for your attention. And if you have questions, I'd be happy to take them.
Very short question. How did you do your curve fitting?
This is not a question for me. I cannot tell you the technical background in this particular case. I only know that, for example, when we started to generate first curves, first guys actually used vector images which looked really great on the screen, but it was impossible to use them in a large amount of samples. Like above 50 was almost impossible to really handle and to work with. So then they found some way how to export it or how to use bitmaps, and this is what we have there. I don't remember really the tool for curve fitting algorithm.